Hello friends, my name is Rizwan Hafeez and today we are going to talk about another very important topic, that is complement system. We will talk about the activation of complement system and how this system basically increases our immune response against the foreign particles, against the microbes. Well, first of all, let me tell you what is complement system basically. Complement system is a system of different proteins, approximately 30 different proteins that are synthesized in liver, macrophages, and other immune cells, and they play their important role in increasing our body immunity, in increasing our immune system. How they increase our immune system? They basically increase the phagocytic capacity. They increase the phagocytosis by the phagocytic cell of the microorganism. We know that our immune system had different phagocytic cells like neutrophils and macrophages. So these proteins, complement proteins, they basically increase their phagocytic capacity of the phagocytic cells. In this video, I will be talking about their activation and detailed mechanism, how they basically increase the phagocytic capacity and how they help the immune system will be discussed in very detailed manner. You will be enjoy this lecture. So stay with me. As I have explained that I will talk about the activation of the complement system as well. Well, there are basically three different pathways of the complement system activation. This system of protein get activated by three different pathways. The one is classical complement pathway, the other is alternative complement pathway, and the third one is lectin pathway. In the classical complement pathway, antibodies are involved. And we know that when antibodies are involved, the system will be, immune system will be basically adaptive immunity. It will not be innate. If you want to learn more about adaptive immunity and innate immunity and their cellular aspects, you can watch my video on this topic, adaptive and innate immunity, to refresh your knowledge. Link has been given in the description. Clear? While alternative pathway and lectin pathway, they belongs to innate immunity. Clear? Today, in today's video, I will talk about the classical pathway, and I have explained that in classical pathways, antibodies are involved. It means this system, the system of protein, the system of different these protein will get activated after the antibody and antigen interaction. Now, point is there are different types of antibodies. IgG antibody, IgM antibody, IgA antibody, IgE antibody, and IgD antibody. There are 5 different types of antibody. Now, which antibody will activate or will cause the activation of this system of protein, this complement system? Answer is only IgG and IgM antibody. They have capacity, they have tendency to activate the complement system. Other antibodies cannot activate the complement system. Now let's talk about the detailed mechanism of activation and their significance in the immune system of the classical complement pathway. Now I am going to talk about the classical complement pathway. So here let me make a foreign particle. So for example, this is a foreign particle, or you can say it's a bacteria, and on the surface of bacteria certain antigens are present. For example, these are the antigens, these are the proteins antigens against which antibodies has been formed. Okay, assume that these are the antigen against which antibodies has been formed. Now antibody will come and attach with these antigens with their fab portion. We know that antibody have two regions. This is Fc and they are the Fab. This is Fab and this is Fab. The Fab region is used to bind with the antigen, and after the antibody antigen interaction, this antibody now will be able to attach the first complement protein, that is C1, with its Fc portion. When this antibody was free in the blood, it was unable to attach C1 with this Fc portion. But right after the interaction of antigen and antibody, there will be conformational change in the Fc portion, and now Fc portion will be able to attach C1 along with it. Now, after the attachment of C1 complement protein with the Fc portion, this C1 protein will get activated. Okay, and its protease activity will get activated. Now, this C1 protein will be able to cleave. It will be able to break down two other complement protein. The one is C4 and the other is C2. This C1, it will break C4 into C4b and C4a. Similarly, it will break C2 into C2b and C2a. Okay, the B portion will form a complex and A portion will stay away. We will talk about the significance of A's and B's in later part of the video. Stay with me. Okay, now B portion will make complex. So this complex C4B and C2B, first C4B will attach and then C2B will attach. Okay, C4B and C2B, this complex is basically called as C3 convertase. This complex is called as C3 convertase. Now, if you look at the word C3 convertase, it means this complex will be able to convert C3. Now, this complex will convert C3 into C3A and C3B. Again, the B portion will make complex with the other proteins, and C3A, it will stay away. We'll talk about the function of Ca in the later part of the video. Now the complex C4B, C2B and C3B. Now this complex C4B, C2B and C3B, this complex is basically called as C5 convertase. C5 convertase. It means this complex of three protein will be able to convert C5 into C5A and C5B. Now C5B will it will not attach, in fact it will stay separate. Now C5A and C5B has been formed. Now some other complement proteins which include C6, C7, C8, C9, they will attach with the C5B and a complex will be formed. This complex of 5 proteins, 1, 2, 3, 4 and 5, 1, 2, 3, 4 and 5, this complex of 5 protein is basically called as MAC, membrane attack complex. It means this complex is going to attack on the bacterial cell membrane. Now let's see how it will attack. These proteins, all these 5 proteins, they will arrange themselves to form a pore-like structure, to form a hole-like structure. In this way a pore will be formed. Okay, all these 5 proteins, they will arrange themselves to form a pore. Now this pore will be placed on the bacterial cell membrane, and once this pore will be placed on the bacterial cell membrane, obviously the permeability of the bacteria will get disturbed. So it means all the required component All the important components, they will start leaking out of the cell, and undesired or harmful substances will start moving inside the cell, which will result in bacterial death, and ultimately bacteria will lyse. It will be broken down. It will be lysed. Okay, so this is the first function of this complex MAC, or these complement system. First function, cell lysis, or microorganism or pathogen lysis. Okay, now let's talk about another very important function. This C3b, here I'm going to draw another bacteria because lots of protein has been made over, so it will be very difficult for you to understand the exact concept. For example, this was our bacteria, and here it was our antibody, and the first complement protein was C1, then it was C4b, then it was C2b, then it was C3b. This complex was formed with the Fc portion of antibody. Now, in the definition, I explained that these complement system, these complement protein basically increases the phagocytic capacity of the phagocytic cells. How it increases the phagocytic cell? This complex basically act as a sweet candy. It attracts the phagocytic cells like neutrophil and macrophages. How it attract? For example, here let me show. For example, this is a macrophage that is going to eat up this bacteria, this foreign particle. It will be having receptor for this C3b on its surface. Okay, now this C3b will attach with these receptor. Now the whole complex, C3b, the complement complex, antibody, and the bacteria. Okay, the bacteria is basically attached with the antibody, no? In this way. Okay, now this complex has been trapped, it has been held tightly. Now it will be very easy for this macrophage to engulf this uh, microorganism or bacteria. So in this way, these complement proteins basically increases the phagocytic capacity of the phagocytic cells like neutrophil or macrophages. Assume that this is macrophage. Now, after this attachment, it can easily engulf or eat up this bacteria, and it can destroy it into the small fragments. Clear? So, in this way, the antibody can also directly, but forget about it. In today's video, we'll focus on the complement system. The role of antibody will be discussed in some other videos. Now, let's see the role of these A portion, why they were formed, and what is their role in the immune system. Okay, as we have explained that proteins, all the proteins, this complement protein, antibodies also protein, so proteins and some enzymes also, so proteins have very important role. Okay, now let's see how this A portion is going to play their role. First of all, let me tell you, these C4a, C3a, C5a, they are also called as anaphylatoxins. They are also called as anaphylatoxins. Anaphylatoxins. What do you mean by anaphylatoxin? Anaphylatoxins are those substances which can cause the anaphylaxis. Okay, which can cause the release of histamine from the mast cell, and some inflammatory reaction can start. How? This is the infected tissue. Assume that this is the infected tissue, and all this reaction is happening in the infected tissue where the bacteria was present. Okay, now inside the infected tissue, obviously mast cell will also be present. Mast cells are very abundant in the skin, inside the lungs, inside the GIT. They are very abundant. So these mast cell, they will be having receptors for C3a, C5a, and C4a. And once these A portion, they will go and they will attach on these receptors inside the mast cells. You know that mast cells are granulocyte. They are not agranulocyte. They are granulocyte. So they will be having small granules in which preformed mediators like histamine will be present. So once these uh, proteins they will attach on their receptor, mast cell will release the component of these granules like histamine. Okay, now histamine has been released. Now what will happen? Histamine will be having receptor on the smooth muscle of the nearby blood vessel and on the endothelial cells of the nearby blood vessel. So for example, they are the endothelial cells. Okay, endothelial cells. When the histamine will act on the smooth muscle of the nearby blood vessel, it will cause a vasodilation. So more blood will come toward this side. And why we need more blood? Obviously because when the blood will be more, the nutrition, the protein, the antibody, all these complement proteins and even the leukocytes, they will come more toward these sites which are required because the bacterial infection has happened on these sites. So we need more blood flow, and this thing will be done by the histamine. Histamine will cause the smooth muscle relaxation of the blood vessel, which will cause the more blood flow. And at the same time, this histamine, it will act on these cells, and it will also cause the shrinkage of these cells. When these cells will get shrink, contraction of these cells, the gap between the cells will increase. Here you can see the gap between the cell will increase, and once the gap will increase, the leakage of the fluid and complementary protein and other protein and antibodies, it will increase. Okay, so it is also basically helping our immune response. So this is the function of a portion. Some other very important function is the C5a. C5a also have receptor on these vascular endothelial cell. When the C5, it will go and it will act on these receptor on the endothelial cell, what will happen? Some important proteins will appear on the endothelial cells once the C5a will bind to its receptor on the endothelial cell. These important proteins are basically responsible for attaching, they are responsible for catching, for adhering the neutrophils passing through the blood. These proteins, they will catch these neutrophil, adhere these neutrophil, and drag them out of the blood vessel inside the tissue. Now neutrophil will be transmigrated inside the tissue. Okay, so it means C5a is basically enhancing the transmigration of the neutrophil from the blood into the infected tissue. Now it is also going to help in the chemotaxis. For example, inside the tissue, this is the area where the infection has been taken place. Now this C5a, it will also help the neutrophil. It will also help, sorry, neutrophil. It will also help neutrophil in moving toward the site of infection, chemotaxis. So this will also be done by the C5a. So these are the function of A portion as well. We have discussed the function of B portion and A portion as well. One important function I would like to talk about is this C3b. It also have receptor on the B lymphocytes. We know that in the nearby lymph node, for example, this is a lymph node, B lymphocytes will be present, and when this C3b will go and it will act on the B lymphocyte, C3b, when it will act on the B lymphocyte, it will increase the proliferation and differentiation. So once the proliferation means division, more and more copies of B cells will be formed, plasma cells will be formed. So obviously plasma cells are responsible for making antibodies, so more antibodies will be formed to tackle the infection. So here you can see that how these complement system is basically increasing our immune system. It is increasing the antibody production, it is transmigrating the neutrophils, it is dilating the blood vessels, it is causing the opsonization, this phagocytosis, the increasement of the phagocytosis is basically called as the opsonization. And they are creating the MAC protein, which is directly causing the lysis of bacteria. So this is all about the complement system and their role and their activation through the classical pathway. I hope you like my video. Please don't forget to subscribe my channel and press the bell icon. In the next video, I'll be talking about the uh, alternative pathway and lectin pathway and their activation and their mechanism will be obviously approximately same, but their activation would be a little bit different. I will talk about that in the next video. Stay with me. Thank you so much.